This is the public lecture series, for those of you who are new, is something that we've been doing for the past 11 years. We use this as an opportunity to educate the public about marine science, but also to highlight the work of our own researchers. So most of the time, the people that are speaking here at our public lecture series are our own scientists or our graduate students, but occasionally we will bring in people from other places. And tonight is one of the very special nights that we have every year. It's a night when we bring a distinguished guest in from another university, being supported by an endowment that we have called the Laura Randall Schweppe Endowed Lecture Series in Marine Science. And so that gives us the opportunity to bring accomplished scientists to the Marine Science Institute to work with our students and our faculty members to further research and to exchange ideas. And we try to twist their arm to talk to the public about their research as well. So it brings you another dimension of what's going on. And I'll tell you a little bit more about that in a few minutes, but I want to get some other things out of the way first. Those of you who have been coming regularly know that we're getting toward the end of our series for this season, and I appreciate those of you who have joined us week after week. As you can tell, the numbers have thinned a bit since we had 260 or so people here earlier in January. But we do have one more talk coming up next week, and instead of Thursday, it's going to be on Tuesday night. And this is another special occasion of a visitor brought in by the same endowment that tonight's visitor is here. And that's going to be Dr. Steve O'Brien, O'Brien, who's an incredibly accomplished scientist, and I hope you'll have a chance to stop by and listen to that talk as well. As usual this year, our talk is being recorded, and we put it onto a DVD and send it over to the Bay Education Center in Rockport so that people can enjoy this talk afterwards or during the next week. So if you have friends that couldn't make it tonight and you want to tell them what a great time you had here and they have a chance to see it, they can go to the Bay Education Center next week. Finally, I've started posting these lectures on the University of Texas YouTube site, so you can actually watch it from the comfort of your home as well. And if you want to know when those are posted and where they are, you just go to our Facebook page, facebook.com/UTMSI, and that'll tell you how to get a hold of these things. Okay, so one of the things that we always, by tradition, start our public lecture series with is this week's episode of Science in the Sea. Science in the Sea is the University of Texas Marine Science Institute's radio program. It's a 2-minute excerpt on marine science. It airs on 182 radio stations around the country in about 40 states, as well as American Forces radio network around the world. So let's have a listen to this week's episode of Science in the Sea.
Exploring science in the sea. For a young fish, moving too slow can get you snapped up by a bigger fish. But so can moving too fast, or even being too timid. When small fish that live on coral reefs hatch, they stay near the surface, generally following the currents. When they get older, though, they settle on a nearby reef. But most of these new arrivals get eaten within a couple of days. A recent study found a few common traits among the fish that are the first to go. Led by University of Texas marine biologist Lee Fuman, the study examined young damselfish on Australia's Great Barrier Reef. In the lab, researchers measured how fast the fish swam and how they responded to possible predators. Then they released the fish on small patches of reef, and divers measured how bold the small fish were. Finally, they checked back every few hours until half of the test subjects were gone, most likely because they were eaten, about 24 hours. The first to go shared several traits. A slow response to danger was one. Another was that they were fast swimmers, perhaps drawing attention to themselves as they skittered around the reef. And third, they were shy. They were quicker to hide, and they stayed in their hiding places longer. The researchers suggest that shy fish may not be as aware of their surroundings as bolder ones, so it's easier for predators to sneak up on them. But it was harder to figure out which traits offered the best chance of survival. There's no sure formula, it seems, for surviving the first few days on a coral reef. This episode of Science in the Sea was made possible by a grant from the National Science Foundation. I'm Holly Brawley.
Well, I hope you enjoyed that. That was one of the few times it was about my own research. Okay, let me turn the lights on here. Right. What I want to do now is tell you a little bit about this Laura Randall Schweppe Endowed Lecture Series in Marine Science. It was created about 10 years ago or so by a friend of the Marine Science Institute named Irving Schweppe and his daughters. They created this endowment in memory of their mother and wife, who was a lover of the coast, and especially Port Aransas. She had a deep love of this particular place on the coast, and they saw fit to remember her with this gift to the Marine Science Institute. And Dr. Schweppe wanted it to be more than just an opportunity to have a scientist come and give a seminar. He wanted it to have a bigger impact on the students and the faculty members at the Marine Science Institute. And so he encouraged us to have our visitors stay for more than just one day. And so in fact, we require our guests that are funded by this series to spend at least 3 full working days here at the Marine Science Institute. And some of them stay much longer. And this gives us an opportunity to develop research collaborations, follow up on existing collaborations, and of course, many of our guests will participate in our graduate classes, helping the students learn or see new dimensions to the fields they're studying. And it's just been a marvelous, marvelous program, and we've had some extremely outstanding and distinguished scholars come visit us, and tonight is certainly no exception. And when we do have a guest come, they're nominated by either our graduate students or by a member of our own faculty, and that person or group hosts the guest for the time that they're here. So I'm going to have the host of tonight's speaker introduce tonight's speaker, and that's Dr. Ben Walther, who those of you who have been here this season will know is our second speaker this year. So let me turn this over to Ben.
Thank you, Lee, and thanks to all of you for coming. It's spring break and fine St. Patrick's Day evening. How many of you were at the lecture that I gave about fish ear bones? Several. Okay, you're going to hear some even more interesting information about ways that we can use those to ask questions about fish ecology. And so I'm very pleased to welcome Dr. Karin Limburg tonight. She is an associate professor at the State University of New York in Syracuse. She received her master's from the University of Florida and her PhD from Cornell University, and I've known her for several years. I was doing my PhD up in Woods Hole studying American shad, and among many other things, Karin is also an expert in American shad, and I asked her to serve on my committee, which she did, and we've been colleagues ever since. So it is my great pleasure to introduce Dr. Karin Limburg.
Okay. So, first of all, thank you very much for nominating me and for and to the UTMSI for actually having me down here. It's really an honor to come and be one of these lecturers. When you look at the list of people who have come through these doors as a lecturer, it's pretty humbling to be counted among this group. So it's really great for me, and I've also really been curious about this place for a long time, so finally I've had a chance to come down here. So I was going to ask if anybody else here was from New York. Anybody here from New York besides my husband? You're a long way from home, except you've lived here for a while, I guess, right?
I graduated from Cornell.
Well, oh, good, very good. So did I. Okay. Well, we may as well get started with the lecture, and I'm going to just put this over here. Maybe I'll put the water somewhere where I won't spill it. I do tend to wave my hands a lot. So, Linda, if we can dim the lights, we can get started. Put that over there. Okay. Well, I'm going to take you somewhere else tonight. Far, far away, I guess. But this is what I decided to give as my talk title. And so these ear stones or ear bones, some people call them ear bones, but they're really not bone. So I want to see if I can convince you that they can tell us something about this phenomenon called hypoxia. So, and these are dead menhaden back in the background there, by the way. So here's the outline of my presentation, and I'm going to sort of talk about the first 2 points first, and then go into a little sort of side story about these ear stones, which we call otoliths, and then why these things might help us to understand this phenomenon called hypoxia. So hypoxia is defined as conditions that are low oxygen. And some of you might have known about them as, it's a medical condition. Certainly it's associated with COPD and things like that, where you don't have enough oxygen in your blood, but it's not just a medical condition. In the aquatic world, we refer to it as when ecosystems are sort of depleted in dissolved oxygen. It's defined below some threshold value, and sort of functionally, operationally, it's when fish start not doing very well. And so that's about 2 milligrams per liter of dissolved oxygen, about 2 parts per million dissolved oxygen. And it's caused by, it can be caused by different things, but increasingly it's caused by eutrophication, which is sort of the over-fertilization of water bodies. And a good example of this is Lake Erie over here. And many of you probably remember the newspaper headlines in the 60s and 70s about Lake Erie being dead. It wasn't dead, it was just overloaded with phosphorus. And even today, the western basin of Lake Erie is overloaded from Detroit and so on. And so it's, this part is still pretty, I just was reading about it. It's considered quite hypoxic once again now. But Lake Erie was actually responsible for getting the phosphate bans in New York to, getting that reduction down. So, when you get all this nutrient input, then you get big blooms of phytoplankton, the little planktonic organisms, and they, after they die, they fall to the bottom, and then bacteria decompose them, and the bacteria consume oxygen, which is kind of what causes this problem. So then, of course, the one that probably most people know from the news is this big zone of hypoxia that sets up every year now in the northern Gulf of Mexico, around around Louisiana's coast, and this is sort of an image of that too. Draining, the Mississippi drainage is the biggest watershed in the country, and all the use of fertilizers up here in the corn and soy belt, it leads to a tremendous amount of nutrients that come down the pipe, so to speak, and end up fertilizing or over-fertilizing the Gulf of Mexico. So we have this zone that's about the size sometimes of New Jersey and Delaware combined that they commonly refer to as the dead zone. So we have those kinds of phenomena, and with more people on Earth and more economic activity and more mouths to feed and so on, we have increased the amount of nutrients that we're flushing off the land into waterways and then out to the coastal zones. So there are actually now many of these what are called human-induced dead zones around the world. There are some natural ones too. The Black Sea actually has a large area of anoxic water, water without oxygen in it. That's a natural system, but most of these are actually human-induced, and they're on the rise. Now, one of the effects of this, of over-fertilizing the waters, is that you switch your systems from things like, if you think about coastal waters, you switch from these kind of seagrass beds that are adapted to low nutrient regimes. And this is a graph from a paper that was published a while ago that just sort of shows the transition as you increase your loading rate of nutrients, in this case nitrogen. As you increase that, your proportion of seagrass beds like this declines. You may get a shift over to some macroalgae, these things like, I can't remember, Ben, what's codium called in English? Do you remember? Dead man's fingers, thank you. So dead man's fingers or sea lettuce or whatnot. If you really load it on, you get a switch over to phytoplankton-dominated systems. So this is a picture I actually took out of a plane window flying out of Copenhagen, Denmark, and a phenomenal bloom there of phytoplankton, just waves of phytoplankton. They produce a lot, they discharge a lot of agricultural nutrients in Denmark. So that's kind of what happens. And then, of course, this stuff, when it dies, and decomposes, the bacteria decompose it and consume the oxygen. So you really switch those systems around. And so when you think about how does that affect fisheries, it's kind of quite variable, I guess you could say. Fish, of course, being mobile, or most of them are fairly mobile, can get away. They can swim away. And so if they're in a place where they can actually escape, sometimes they're concentrated into some place where fishermen actually have an easier time capturing them. So sometimes it actually makes for better fishing in a sense, although it's not making for more fish. It's just maybe concentrating the fish. If there's very acute, if they're really trapped in a hypoxic or anoxic region, they might just die, fall to the bottom, or you may see something like what's called a jubilee. Has anybody here seen a jubilee? A jubilee is when the fish swim out of the water looking for air, and people just scoop them in and say, hallelujah, it's a jubilee, we got all these fish. And so that's what they call it, actually. And they even occur up in Long Island Sound sometimes. And then you get fish that are, exposed, are chronically exposed, and they're not going to probably do very well. Or they may adapt. Their hemoglobin actually can adapt. And then if we look at, again, sort of a theoretical graph showing nutrient loading, going from, sort of low nutrient dominated ecosystems to very eutrophic to what even is called dystrophic, you see a prediction of what you might expect. So you fertilize these systems to some extent, and they start to, they respond by producing more biomass, more fish biomass, more shellfish biomass, and so on and so forth. But then it's sort of predicted that at some point you're going to kind of go over the top, and it's just not going to be productive anymore, and you end up in situations where you have seasonal hypoxia, or even if it's really, really severe, permanent anoxia even. So we're now going to go over across the puddle across the Atlantic over to Europe, and I'm just orienting you with a map of Europe. I know it's a long way from Texas, right? And so my study is sort of focused on Sweden. My colleagues are Swedish, and just so you know, it's not the same country as Switzerland. I don't know how many times I tell people I'm half Swedish, and then they ask me about Switzerland. You can really see they're different countries. And then this is the Baltic Sea. So this is where my study actually takes place. And the Baltic actually happens to be the mother of all dead zones, or human-induced dead zones. I would say human-enhanced because actually, naturally, it does actually go hypoxic naturally. There have been studies that have shown that over time, since the formation of the salty phase of the Baltic Sea's history, it's gone about 8,000 years ago. It's actually had periodic dead zones. But, so it's a very large dead zone. And just for scale, just to keep this in mind, that Sweden's about the size of California. So this is a really big, big area, right? The black is where there's no oxygen at all on the bottom, and the gray is where it's less than this in this case, 2 milliliters per liter. So here's the star of tonight's talk is the cod. And the cod is a fish this is the same species as the North Atlantic cod that we see in New England and so on, but it's genetically somewhat different. It's been here in the Baltic. Certainly this I think it was, here you'll see some of the evidence of its prehistoric time here. It disappeared for a while and then came back again in the Middle Ages. But the Baltic is an interesting semi-enclosed sea because it's actually fairly fresh up towards the north in the Bothnian Bay, and has a sort of a stable, more or less stable salinity gradient out down here to the outlet to the North Sea. And its average salinity is about 8 parts per thousand. But the cod actually require, for their eggs to be fertilized successfully and to hatch successfully, they require salinities of about 11 parts per thousand, which only occurs at depth in the Baltic Sea. So unfortunately, these deep places, this is a deep place and this is a deep place, these are actually deep places too, and these are unfortunately places that have low oxygen today. And so if you look at those 4 areas there, those colored hoops, and you map them onto a map from an article about spawning and nursery habitats for the Baltic Sea cod, it's not a good picture, is it? That, in fact, the Gotland Basin is pretty much gone now as a spawning habitat, and the Gdansk Deep also is not often available, so to speak. So really, most of the spawning these days takes place in the Bornholm Basin. So, okay, so that sort of sets the stage there. Cod interact with this hypoxia, we think. We know that they do, actually. So now I'm going to take another tack and tell you a little bit about earstones, or otoliths. And take a little water. So I guess Ben already told some of you about otoliths, but the rest of you might be wondering what they are. And they certainly are funny-looking little things. Well, what they are part of the hearing and balance system in fishes, and they are, and actually we have little crystals that form in our ears too that are not quite the same. But anyway, all the modern fishes have these otoliths, and they come in 3 pairs: a big pair that's called the sagittal otolith, a very, very tiny one that's called the lapillus, and another one that's sort of a funny shape called an asteriscus. And this is how they sit in the semicircular canals in the heads of fishes, and these things sit under the brain case. And these walls of the little sacs that they're in they sit in a fluid, and so that they can rock around as the fish moves. And the walls of the sacs are filled with nerve cells. And so as the fish moves around, these things sort of rock and hit against the walls of of these chambers, and then hit nerve cells that then fire off signals to the brain and give information on inertia and direction and so on, and sometimes they're even involved in hearing. So that's an important thing, but why fisheries biologists and fisheries scientists are really fascinated by them is because other properties I'll get to in a moment, but I just wanted to show you a picture from my own private collection of earstones that they come in many sizes and shapes. And these are adult ones here from herring. This is an adult cod. This is actually quite an old one. They get kind of scalloped and sculpted when they get old. Let's see, these are striped bass. This is carp. This is a freshwater drum. I think that's a snapper. No, that's snapper there. Anyway, lots and lots of different shapes and sizes. They're really kind of interesting looking. But what we like about them is that you can actually slice them. These are pretty small. They're quite small, but you can slice them up and then look at them under the microscope. And when you do that, so this is out of a little 20-millimeter larva, so that's not quite an inch-long fish. And somewhere in its head here is a pair, or 3 pairs of these tiny otoliths. And when you pull them out, you can actually see daily, look them under a microscope, and you can count these daily growth rings, which is pretty amazing that you can actually get the age of a fish to a day. But also you can see information at an annual scale too. So here's a salmon head. I was dissecting some earstones out of them. So there are the earstones sitting there. I took the brain out. And so we have nice annual bands that you can see as well. So pretty remarkable. And embedded in these, if I removed the overburden, I could see the youth of this fish as well. I could see those daily growth rings in its young stage. So I made this sort of schematic for one of my colleagues to show him how I was preparing earstones out of cod. And so here's a cod otolith, and I embed it in some plastic, and then I have a saw, a very fine saw that I use to cut out a section, and then I have to thin it down to maybe a couple of, thickness of a couple of pieces of paper or something. And then you can see these growth rings, and it's not so hard to count these things. in some of the populations. So it's really been, now it's quite standard practice for us to use these to do age and growth studies, and it's very important for our understanding of the demography of fish populations. So we call them maybe biochronometers or the Rosetta Stones of fish. Often we sometimes call them the CD-ROMs of the store information about fish that we're trying to unlock. I don't know what you called them, Ben, but did you call them something like that? No, I think, or the black box in a jet, something like that. Well, it turns out that we can also look at the chemistry of ear stones, and they are not, as I said, they're not bone. Bone is a mineral called apatite, which has phosphorus in it, but these are calcium carbonate, and it's deposited there's three forms of calcium carbonate. The one that you might remember from your chemistry class is called calcite, but in ear stones and in corals also, the formation is mediated by enzyme reactions, and so it actually precipitates in a special form called aragonite. And if it's laid down every day, a little bit's precipitated every day as on a protein sheath that sort of grows out a little bit, and then this stuff, the calcium carbonate precipitates on that. And so what happens is that a number of trace elements, and a trace element just means it's not much there, and then isotopes are taken up from the environment and they incorporate into ear stones. And so we've started to take advantage of that, and more people are interested in this. So we can use a number of different instruments to quantify these kinds of chemistries. And so one of the elements that has been really, really useful to look at is strontium. If you look at the periodic table, I know you're all going to go home and look up your periodic table of chemistry, right? So if you look at where calcium is, and you look right below it, is strontium. So they're in the same sort of family, the alkaline earth elements in the periodic table. And it's very close, its radius is very similar to calcium, so it can actually substitute, that atom can substitute into the calcium carbonate molecule pretty readily, which is pretty cool. So, and it turns out that Howard Odom, who was not only the third director of this august institution, was also my master's advisor, but we did completely different things. I wasn't doing any of this then. But he studied, he did his PhD at Yale, and he studied under a very, very famous limnologist named G. Evelyn Hutchinson. And Hutchinson had him work on the biogeochemistry, global cycling of strontium, the element strontium. And Odom had published a lot of that work while he was here. So these, we've been looking at some of Odom's publications in the library here, but he wrote some wonderful, wonderful papers about his work, and he collected data on everything, and he even looked at ear stones of fish, looked at rocks and seashells and ear stones and stuff. And he noted that, among other things, that this ratio of strontium to calcium, we often take a ratio of these things to sort of control for variation in calcium, was higher in marine fish otoliths than it was in freshwater. And this is because there just tends to be, relative to calcium, there tends to be more strontium in marine water than relative to many, many freshwater systems. Not all, but many. And the other thing about Odom that was remarkable reading his papers was that he actually built his own instrumentation. He built his own spectrophotometers. And you read his papers, and today nobody would dream about doing something like that. They'd just go to a catalog. They'd try to write a $500,000 grant and get something off the shelf. But this guy was really remarkable. Now, not everything is still, is just off the shelf. I've actually worked on a homemade system myself. I didn't make this, but some nuclear physicists made this instrument. This is in southern Sweden at Lund University. I was at Stockholm University before my current job at SUNY ESF. But this is a kind of an instrument, you can see the name up here, a big name. We call it just micropixie for short, but this is actually a small mass accelerator. It's only 15 foot long or so. And then, so you accelerate mass, and we shoot a beam of proteins down this beam line. And somewhere out here, I have there's a little vacuum chamber, and I have put my little otolith samples in there. And so this beam rasters along, and we can with this instrument, we can make maps of the location and concentrations of some elements. So this is an otolith of an Atlantic salmon. And what we see here is an elemental map of strontium and another one of zinc. And the first thing that you notice is that they look different from each other, right? The patterns are very readily apparent here, and they're very different. And this actually looks kind of more like the optical image, doesn't it? But anyway, so strontium, just very quickly, tells us that there's this blob of strontium in the middle. Turns out that mom, where she's developing her eggs, she picks up the strontium signature and she puts that into the egg. So that's the first thing that you see. And so we can see that that mom put her strontium in a place that was different from where this fish actually reared. It reared in a very low strontium place, and then it went into a very high strontium environment, and then kind of went into something a little bit lower again. And I'm going to tell you, it looks a lot like a marine system, but this is actually upstate New York. And this is the Fish Creek that drains into Oneida Lake, which is an entirely inside New York State lake, but it's loaded with strontium. Why? Because it's sitting on ancient marine bedrock that is loaded with strontium. So I like to call it the Sea of Oneida instead of Oneida Lake. But anyway, so we can get really cool information about how long this fish stayed in Fish Creek, a couple years, and then moved into some part of Oneida Lake that had a lot of strontium, and then moved out into something that was a little less. And the zinc is following annual patterns. It's actually higher in summer and lower in winter. But let me also tell you that this has nothing to do with pollution at all, not a thing. So it's no pollution here. Okay, another instrument that's kind of homemade too, I guess you could say, is a much, much larger one. This is underneath the Cornell football field is something called a synchrotron. And some universities, I don't know if the University of Texas has a synchrotron or not, but Cornell's only an hour away. And so they built this thing to do high energy physics experiments, but it generates a lot of high energy X-rays. And in about the 1970s or 80s, physicists started to use those X-rays for other experiments. And they do a lot of work. They found that they could do a lot of applications with the X-rays. Among other things, one of the things that they do an awful lot of is they characterize protein structure there. So a lot of biomedical applications come here and do that. I think they actually characterized the rhinovirus here, the one for the common cold, at Cornell at the synchrotron. This building here is where the scientists are, and it's the acronym here stands for the Cornell High Energy Synchrotron Source. And so it's a big building which has offices but also labs and then getting into the synchrotron. So this big ring goes under the football field, and this is a Nobel Prize winning physicist named Hans Bethe, and he was one of the people who helped to build it. And here he's just demonstrating how big the ring is by riding a bicycle through it. Obviously, it's not in operation here because you wouldn't want to be in there when there's lethal doses of X-rays going on. But I've actually walked around in it when they've had it down for servicing in the summertime. It's pretty interesting. And the place where I work is this little nook back here in the basement, one part of the basement. It really is like being in Das Boot, in a U-boat, just sitting down here for a week at a time. And there's a chamber in here. This says the F3 hutch. It's the station there. And it's got heavy lead shielding all over the place here. And when you go inside, you can't go in when the X-rays are going because you would die. But you can, they have lots and lots of safety features so that you will not kill yourself there, which is a good thing. And but but when the beam isn't going there, then you can go in and set up your sample. And we set up our sample on a little, just take a little, a slide from a regular 35 millimeter slide, just take the holder, and then we can put our otoliths, we sort of tape them in and then put them in another little holder. And this is a camera here just to visualize it. And there's actually a special little glass capillary that takes this high energy X-ray and focuses it very, very precisely. And we can focus it down as small as about maybe 10 microns, which is very, very, very small. And then there's a detector here. And what we're doing when we hit the sample with X-rays is, it's kind of similar to the map I just showed you. We excite the atoms on the surface of the sample, and that kicks electrons off. And as electrons are kicked off the atoms, others from the upper shells sort of fall back down in, and that releases energy. And every element has sort of characteristic fluorescences from that. And so you can, if you know what those are, you can quantify the concentrations. And so this is a detector here that does that. And we had to use the fastest detector in the house because we generate so many counts. So what do we get? Well, the first time I did this, I went down with a whitefish otolith from the Baltic Sea, a species of whitefish. And the Baltic is a funny place. It's sort of intermediate between fresh and marine. And yet the organisms there sort of seem more or less marine, I guess. I have to, truth in advertising here is I actually crack my otoliths sometimes to flatten them out. So you do see the cracks here. But here's the optical image, and then here's an image of calcium, which is pretty homogeneous. And then here's strontium, and the big thing to see here is that kind of like that salmon we saw before, this, and whitefishes are related to salmons. It actually has low strontium in this inner part for about 1 year's worth of life. And then it moved into a higher strontium environment. And we know in the Baltic Sea that this is freshwater, low strontium is freshwater, and high strontium is brackish or salty water. Conversely to that, there's actually a lot of barium in this inner region, and the soils in the Baltic drainage are full of barium, and the Baltic Sea is rather low in barium. We also see zinc, again, this sort of funny configuration of zinc, and what I learned was that zinc in this group of fishes, the salmons and the whitefishes, and actually even the pike family, actually accumulates zinc in this sort of funny way, so it seems like a genetic effect. So it's kind of interesting. But then here we have manganese. So we have these really distinctive bands of manganese that actually are showing up in the same sort of bands as these high zinc bands, which means that it's summertime. So when I saw this, I thought, I wonder what's going on here. Okay, but I digress a little bit. Back to codfish and to hypoxia. So why these earstones might help us understand hypoxia. Well, to talk about this, I'm actually going to go back in time just a little bit. It turns out that, as I say here, cod have been around in the Baltic for a long, long time. And this goes back to the Neolithic period. And while I was at Stockholm University, I actually met and started working with a doctoral student who I ended up co-advising in a field called osteoarchaeology, so the archaeological study of bones. And she did a really great dissertation on fisheries of the Neolithic period. And so here we have one of the really well-preserved skeletons of people from this one place, and here we have piles and piles of cod vertebrae. So where we do our work is on the island of Gotland, which belongs to Sweden. It's Sweden's largest island in the Baltic, or anywhere, I guess. And this is a map published from a paper about archaeological remains on Gotland. And so it's showing us the settlements. So all these dots are where the archaeologists have found settlement remains, evidences of settlements. And the dashed line out here is the modern coastline. And the Baltic Sea was higher relative to land a few thousand years ago because the land was still coming up after the glaciers melted. So there's what's called rebound after that. And so they were still depressed relative to what they are today, more or less. And these zones around here are rings that the archaeologists drew to show what the sort of the territory, the range would have been for people, where people would have gotten their food and their other resources from. It's about 6 miles in radius. And so, you know, settlements in the middle here would have had mostly land-based, maybe lake also based resources, but those on the coast are gonna have, you know, a mix of land-based and marine resource use. And this site here, which is called Aveda, had clearly a predominance of marine resource use, and that's quite evident in the remains there of the fish bones and the marine mammal bones and so on. So our challenge was to learn something about the Stone Age use of cod and compare that exploitation to the modern overfish situation. And I actually just gave a seminar about this the other day in a little bit more detail, but just to give you a couple of highlights about that, my student has the arrow pointing at her, Karina Ulsson, and here she's supervising students who spend their summers digging around in this site and just very carefully sifting dirt into buckets, and then they go back here to these sieves and they sieve out stuff, and then they go take their sieved material into a tent and they bag and they tag and they identify stuff. And among other things, they found these preserved otoliths, these ear stones from cod. And these things have been dated with radiocarbon to between about 4,500 and 5,000 years old. So, and although some of them aren't in such great shape, some of them are actually in remarkably good shape. So here's a cross section I made through one of them. And a lot of these, you could actually count these age rings in, right? So we could analyze, we could start to do some demographic studies on these things. So we ended up recruiting an expert from the Swedish Fishery Bureau to do the age determinations. It was somebody who I knew who actually does that kind of determination for the modern stock assessments that they do in the Baltic. She doesn't do it anymore, but she is one of the experts. So we were very fortunate to be able to recruit her. And she always tells me that aging Baltic cod otoliths is generally a black art. This is an unusually nice one. You can really see rings on it. But a lot of them, you're squinting at them and, yeah. Anyway, one thing that we found was that the cod fishery on Gotland 4,500 years ago was probably a nearshore fishery for the most part. We know a lot about the hooks that they used and the line that they used and so on. We even did strength tests on the hooks and stuff like that. But it was probably a nearshore fishery. And today's fishery, the modern fishery, so we did a comparison of 2 recent years after a big collapse. And what we found was that this fishery, which was probably not exploiting the total population all that hard, these fish were bigger than the fish that were, the largest fish that the fishermen could catch today. They're going after as large fish as they can, and the sizes of fish have actually gotten smaller. And this agrees with a lot of other studies that have been going on that have documented the same kind of thing, sort of a response to exploitation. So that's been, that was one thing that we found. This is called, there's actually a term for this, they call it juvenescence. Well, anyway, so when I was messing around with these ear stones, I thought, okay, maybe I should just try to put one of these things under the synchrotron beam and see what I can see. And I was sort of stunned. Here's the optical image of this one Stone Age otolith, and I was really amazed at how many elements I could actually detect in this thing. So I got my strontium and my calcium, and I see that there's sort of lower strontium in the core region of this otolith, and then actually it gets quite a bit higher. So red is high, and I guess sort of the blues are low in this scheme, and most of my other graphs. And so I see that there's low strontium, kind of like, I guess, those salmon that we saw before. And then we see that it gets higher, and then maybe it gets lower on the edge again. I also see bromine, which is almost never reported in fish ear stones. And I even see gold. I know I should have handed out periodic tables for everybody tonight, I'm sure, but I'll just try to explain. So this is barium. There's a band of barium, and typically it's very hard for us to measure barium in ear stones with this method. But we can actually see a thin band that also lights up in rubidium and also lights up in potassium. And it makes me think that perhaps this was overwintering in sort of a freshwater, or not a freshwater, but closer maybe to the mouth of a river or something. And we also, you may have to squint to see this, but there is also banding in manganese in this thing. So I did some more of these, and I was really sort of amazed at the sort of the consistency in these two elements, in manganese and strontium. I kind of thought I was seeing similarities, that I was seeing, sort of a low region of strontium followed by a high region, and then maybe some variation after that, maybe some various banding and so on. And then also banding in manganese. Well, these are modern ones, today's, and these are Stone Age. And I was scratching my head trying to figure out what's going on here. So why are there these distinctive patterns in strontium and manganese? Well, I think we know a lot more about strontium than we do about manganese in otoliths. We know from a lot of experimental work that it varies both with temperature and salinity. It's there, the uptake is a function of what's out there in the water, the amount of strontium relative to the amount of calcium, but it's modified, it can be mediated by the temperature of the water and by the salinity of the water. And we know for cod, for example, there have been experiments done on cod that showed that the strontium to calcium ratio is inversely proportional to temperature, so the higher the water temperature, the lower that ratio is. But from my work here in the Baltic, I think it's also affected by salinity. But anyway, with respect to manganese, people have tried experiments and they have failed to show similar kinds of relationships. So people have sort of been waving their hands about various things. But what we do know is that manganese is involved in what's called redox chemistry. So in the world of biogeochemistry, the chemistry of reduction and oxidation processes. And if you think about, sort of the simplest thing I could think of to describe is if you think about a nail rusting. So the iron in the nail rusts because it's oxidizing. It's being exposed to oxygen, and it is actually forming a form of oxygen that is basically forming this precipitate, this rusty precipitate. And reduction would be sort of the opposite of that. So if we go back to the Baltic again, to these dead zones, if you put, I don't know, this would be an interesting experiment to try, to take a rusty nail, put it down in the anoxic water and see if it unrusts. But basically, you're putting it, these are reducing environments here, okay? So, so they're very strongly reducing environments. So one question then is there a possible mechanism then for the uptake? How does that manganese get into the otolith? Is it governed by these kinds of things going on? And so what we do know is that most of the trace elements that go into otoliths are taken up, they go through the gills as the fish drinks water or breathes, and gets into the bloodstream and eventually into the endolymph in these semicircular canals. And we also know that the dissolved form of manganese is it's a reduced form. It's got a valence state of 2. There are some that are also valence state of 3. The chemistry of manganese is actually quite complicated, but we do know this. And so then the question is, manganese ends up, we do know that there's a lot of manganese in the sediments of many places, including the Baltic Sea. It's got a lot of manganese in the bottom. And when it's exposed to oxygen, it actually forms, you can see places where there are manganese nodules that have precipitated out. But how does it go from the sediments up into the water? And I read a paper that I thought did a very nice job of sort of explaining this. So if we have oxygenated water, the biogeochemists like to call it normoxic water, but it's sitting over sediment. So you have this sort of sediment and water interface. And so when it's oxygenated, you've got manganese oxides that form, and I made a sort of a very simple sort of animation here, but you've got some sediment that's aerated and oxic, and then below that, there's no oxygen. It's been used up basically by the organisms in the oxic zone. And so you actually get sort of a cycle going from sort of this oxidized form to a reduced form, but it just sort of sits here and spins around within the sediments. And this happens typically in seasonally hypoxic zones. It happens in the spring, fall, and winter. And there's some that gets buried into deep sediments too. Okay, so that's what we call internal cycling. But when you have hypoxic or anoxic situations happening, then you have, your oxic sediment may be very, very diminished. It might actually not even be there at all. And so then you now have sort of a situation where you can get this reduced dissolved manganese pumping up into the water column. And so now you have a mechanism to get that stuff up in there. So, and that happens in these seasonally hypoxic areas, typically in summertime. So this is what this kind of sediment looks like. So this brownish layer here is oxygenated. So it's actually, you can actually see rust. There's some iron rusting out right there. And this is all the reduced sediment. It all looks black, and there's hydrogen sulfide in there, and manganese and iron and probably phosphorus and stuff. And if in an anoxic situation, there wouldn't be any of this brown layer here at all. It would all be black. So just to give you a look at what that would look like. So then one question is, how does this enter the otolith? We have a mechanism to get it up into the water column, and it also turns out that manganese doesn't actually, when it hits oxygenated water, doesn't actually just precipitate immediately. Iron, if it gets into the oxygenated water, will just precipitate out essentially like rust and not be dissolved anymore. But manganese sort of hangs around for days at a time. So we know that there's a mechanism to kind of get it up there into the oxygenated water, but how does it actually, enter the otolith, and is it governed? Well, we got the hypothesis that it is taken up over the gills. Is it physiologically governed? I think this still is something that we need. It's a research question still. But here I was trying to figure out, what are these patterns telling me? But I was thinking a lot about this redox stuff. And I thought to myself that it would be really worthwhile. I saw some very strong patterns, especially I was working actually on my modern otoliths with otoliths that were collected in the early 2000s, sort of 2003 to 2006 and so on. And I was seeing a lot of manganese in sort of the core regions of these little ear stones. And I thought, well, maybe I should go back in time, see if I could find some ear stones from like a period when there might not be as much hypoxia in the Baltic. So I didn't know really when that would be, but I thought it might be the 1980s, so I asked my colleague to send me ear stones from the 1980s, and they weren't that much different, to be quite honest. But then, as I was analyzing otoliths, I happened to stumble across a paper that described the time trends and mechanisms of the formation of these big dead zones in the Baltic. And from that, I actually learned that there was a period in the early 1990s that didn't have a whole lot of hypoxia. And so this is that Gotland deep basin that I said is sort of, for spawning, it's a write-off anymore. But a lot of the Baltic actually had well-oxygenated bottoms, all these spawning areas and so on, and nursery areas. And you compare that to 2006 when things are pretty bad, and it's just been getting worse, actually, in the Baltic. But I thought, oh, wow, okay, Yvonne, you've got to send me otoliths from the early 90s. So she did that. And then what I did was I found, by analyzing these things, I found what I think is a pretty remarkable relationship between hypoxia, as indicated by manganese, and hypoxia as indicated by the extent of it on the bottom. That's how they measure it there, and then manganese. And given that the Baltic is huge, it just sort of for scale, this little embayment down here and its watershed's almost as big as the Chesapeake Bay. So this is a big enclosed sea. Okay, so given that I had very few otoliths and a big place to be in, I was pretty surprised that I actually had a statistically significant relationship between the area of the hypoxic bottom and then this manganese to calcium ratio just extracted from the first year of life of the fish. And for statisticians, this is a weak, not so much of the variability in this trend relationship is explained, 42%, but it's quite significant nevertheless. And my colleague, the biogeochemist who I'm working with on this, said that it's very spotty, that hypoxia events are very, very spotty at any rate. So this is a kind of an interesting thing. And there are a lot of other little detail-y things too, but just I wanted to show you that this young of year, so I also got a young of year cod, so a fish that was born in this, a juvenile that was born that spring. It was caught November, I think, 18th in 2008, and although you can't see daily rings here, I thinned it down and I counted the daily rings. I knew it was born in May. And then I took these maps of the manganese and the strontium, and I used a piece of software I had that allows me to sort of lay down a transect and extract the data. And so doing that, then I could sort of parse out the manganese to calcium ratios and the strontium to calcium ratios. And I could see, in fact, that there was a seasonal peak of the manganese to calcium, which is consistent with this phenomenon of seasonal hypoxia. And I also could see that I have sort of an inverse, a loosely inverse relationship with temperature, that I know it's warmest in the summer, and then it gets cold in the fall and winter with strontium. So I had these two things that can help me explain perhaps what's going on. But then what was really, really cool was I could go back and query my Stone Age fish and ask them about the hypoxic situation. And it turns out that my Stone Age fish from the Neolithic period really have, relative to the modern fish, have relatively low amounts of manganese. And they have, if you also look in the core, and unfortunately some of the cores have cracks, so I had to avoid the cracks to take the data. But basically they were really not much different than the core regions of the fish from this low hypoxia modern period. So very little evidence from these fish from the Neolithic period of hypoxia. But then my colleague, my archaeological colleague, gave me earstones from 3 fish from the codoliths that were from a camp that was from the Iron Age, probably about 1,000, maybe 900 years ago. And I analyzed 2 of them the last time I was at the Cornell Synchrotron, and one of them actually showed this really distinctive band of manganese, which I am calling a seasonal hypoxic zone. Whether that was caused by people or just by natural processes, I couldn't tell you. But we can also see that there, again, over time, there are remarkable similarities in the strontium patterns, which suggest to me that there's sort of constancy in migration pathways. I interpret these lower strontium concentrations in the core region as sort of evidence of being in a warmer, less salty, maybe brackish, more brackish nursery area, and then making directed migrations out into colder, deeper water is how I interpret these. So we get different kinds of information from different elements. So this might be a useful insight in how fish interact with hypoxia, and as it turns out, we can see this in other fish species too. Remember that whitefish. So I'm working with a doctoral student at Queens College on Long Island, George Jackman, and he's studying winter flounder up there in these polluted bays in and around New York Harbor. It's a very cute fish, actually. Here it is shambling along, as he says. But these fish, these flounders, actually not only go on top of the water, on top of the sediments, they'll actually burrow right into the sediment, like this one is doing here. And so that blind side of the fish is actually, where there's a gill flap, is actually in contact with this sediment water, which, in polluted places, it's depleted in oxygen. So George comes to me, and he has this really interesting pair of winter flounder otoliths that he took out of a fish that was captured in Little Neck Bay. And so on its seeing side, it has a normal otolith. And on its blind side, it has this funny little deformed otolith that's sort of bluish green, and it's got little dots in it, and little inclusions. And it's such a rare otolith that we called it the diamond. And so this diamond, we were wondering what's in it. Is that copper? It certainly looks like copper. So we probed it, and it turned out that there isn't actually very much copper at all in this thing. It turns out that these blobs are actually iron and manganese and a little bit of zinc too. And the iron is coming up from this hypoxic or maybe even anoxic water in the sediments and forming these funky inclusions, as is happening with the manganese. So I think that's another very nice sort of empirical substantiation of the hypothesis, or more evidence anyway. And then we looked at some of his other otoliths that were collected in various embayments around New York Harbor. So here's the diamond again. And if I put these on a different scale, you'd see very clear bands in the Arthur Kill. It's a very polluted tributary or river, sort of little river around separating Staten Island from New Jersey. So there actually are bands in here, but they're nowhere near as strong as they are in Newark Bay. And so, the inference, I guess, is that Newark Bay may have some serious water quality issues in the summertime. But there are other species, too, that show this kind of thing. So this is a blueback herring from caught in the Mohawk River. And I think that the strontium is telling me that this fish was born in the Mohawk, and then at the end of the first growing season, it migrated through the Hudson to the Atlantic Ocean. But then it probably came back up and spent a second summer up in the Mohawk, migrated all the way back up. And at least that's my current interpretation. And we actually see bands of manganese in its first year and in its second year, maybe even later on too. Here is another Atlantic salmon otolith, a map of manganese from Oneida Lake. We know Oneida Lake goes hypoxic too. We know it has a lot of manganese in it. So I think this fish may have been sort of stuck in a thermal refuge in a hot summer and spent time in contact with water with manganese in it. And then again, here's our Baltic Sea whitefish. So then just to get into the conclusions then, what we have here is sort of an inferred relationship between dead zones and then the presence of manganese in these ear stones. And yes, it would be a lot stronger if we could test this experimentally. It's a hypothesis, but I think, as I like to say, if it looks like a duck, it quacks like a duck, it walks like a duck, it might be a duck, right? So, but I think we still need to do experiments. So the evidence is there, and it can be picked up from multiple fish species. And so given that, and if we can actually substantiate this more strongly, then maybe with the growing presence of dead zones around the world, maybe we can use fish and their otoliths as sort of mobile monitors and really understand how fish interact with these dead zones. So it could be a new chapter in how we do environmental monitoring too, possibly. I mean, it's kind of an exciting prospect. So I think I'm going to leave it there, and thank you for your time. And if you have questions, I'm happy to answer. Is there a dead zone in other of the Great Lakes? I think that there are parts of Lake Ontario that periodically also get hypoxic, and I'm actually not, even though I'm up there, I don't really study the Great Lakes that intensively. I actually mostly work on the Hudson and the Baltic these days. But you know, Dennis? No? I think, well, certainly there's a small lake between Erie and Huron, that's Lake St. Clair, that I think also has some serious problems like that too. But Superior, certainly not. Michigan has had problems from time to time, but it's really not, I don't think it does, I'm not aware of hypoxic areas there now. But Erie is the one that you hear about now as again, having increasing hypoxic problems.
Is the Baltic like the worst worldwide? Is that why you showed that?
Pardon?
The Baltic Sea, is that the worst? Is that why you showed that?
I guess why I showed it is because I started working there when I was at Stockholm University. So I got intrigued with it. And it certainly is one of the worst. I mean, I think it is the largest, as I said in this talk, it's the largest human enhanced dead zone at the moment. And I don't think that's a title to be proud of. But there are 9 countries around the Baltic drainage, and just getting them to come together to solve the problems has been very, very difficult. You know, most of this is from agricultural nutrients that get sort of washed off the fields and loaded into the rivers and eventually make it out to the sea. So it's just like the Mississippi kind of, yeah.
You talked about verification of your theory. Could you talk a little bit about how would you run an experiment?
I'm trying to figure that out now. Actually, I just submitted a small seed grant proposal to try to figure that out. What I'm going to try to do if I get funded to do this little experiment this summer is I'm going to try to take a tank of Oneida Lake water, because we know that it has manganese in it, and sediment from Oneida Lake, which is full of manganese. And I'm going to try to just seal it off and make it go, you know, over time, the bacteria in the water will just make it go anoxic if I keep it sealed off from the atmosphere. And so we're going to put little ports in the side of this tank and draw out water and just verify that it's going anoxic and that it's developing more dissolved manganese. And then when it's sort of cured enough, we're going to take that water and put it into oxygenated, you know, tanks that are open to the air and have fish in them and see if we can basically get that manganese into the fish. And the other way my colleague at Cornell has thought about doing this is at the same time maybe take some bottom water from Oneida Lake, which we can do. We have a way to do that and put that in the tank with the fish too. And then, of course, unfortunately for the fish, we've got to kill it at the end of the experiment to take the ear stones out. There's no good way. Unfortunately, we can't work on these things without killing the fish. So we'll probably work with brown bullhead because they're pretty tough. So, and we know that they can get manganese in the otoliths. And you know, when in a pinch, you can always make them into jewelry, you know. Yeah.
I remember when I was a biology student, bivalves have annular calcium carbonate.
Right.
Does anybody work with manganese and all that?
You know, I should know the answer to that, and I don't. But it seems like they'd be natural, wouldn't they? And I know that people work with bivalves a lot as, you know, because they don't move that much, so they're ideal biomonitors. And people use them to look at heavy metals, but I have not seen anything about manganese, to be honest. I'm a little embarrassed that I haven't searched the literature as much as I should, but yeah, really. But I'm going to be looking at some more flatfish, and I guess the next step would be probably to at least look at the literature and see if people have been addressing this issue through that. But I have not turned up anything. Anybody else have a question?
I do.
Yeah.
Are the ear stones in all fish the same in size and
Oh, no.
consistency?
No. You have to think about the fact that they have to fit in those skulls, and you think about the diversity of fish and the diversity of shapes and sizes of fish. No, they're all really different. In fact, people can do taxonomy on the shape of these things. There are actually books that are published that show different ones. So herrings have kind of small ones, and I guess the largest I've seen were from I think it was from a fossil fish. It was I think in the drum family, it was actually something like about that big. But the ones I mostly work on are really, really tiny.
So they're collectible.
They are collectible. Actually, a great place to collect is on the shores of Lake Erie and Lake Ontario. You'll find all these freshwater drum otoliths along the shore, and you can just pick them up. And
I started collecting the hardhead, the crucifix fish.
I don't know that one. Is that down here?
Quite beautiful.
Yeah? Yeah, neat. Actually, this guy from Ohio contacted me about 2 months ago. He had a question. He had some of these drum otoliths from Indian middens, I guess, near Sandusky, and he wanted to know if I would analyze some of them for him. And I said, yeah, if you're willing to pay the analytical costs, I'll do the prep and do that work for you. Before I knew it, he sent me his entire collection. I got this box. And this guy collected otoliths like stamps. And so You know, it was, it's a great reference collection for me, but I haven't had a chance to go through it yet. But yeah, people do collect them. And I show a slide when I teach my fisheries class, and I give a lecture about earstones. At the end, I show a picture that a former student of mine took up in Alaska, which was a big billboard that said otolith jewelry on it. So I always say, you know, if you can't get a job, you can make otolith jewelry. So.
There's a question up here, Karen.
Yes.
I don't know what I'm talking about, but do you do anything with mammals?
I don't do anything with
I mean,
mammals.
have they done anything like
Oh, have they done anything like this with mammals? Have they done anything like this with mammals in terms of looking at the bones and looking at elements? Yeah. Yes, actually they have. People do that sort of work. I don't know. I mean, all these different groups of animals are going to have different mechanisms that as they take up elements or isotopes, their bodies are going to do different things to them, I guess. So all I can say is I've seen some studies, and I think some of the chemicals that are studied in mammals' bones are proxies for temperature, I guess, oxygen isotopes, for example. I've seen studies of some other isotopes that give us information about their position in the food web too. Yeah. But so bone is it's nice because it tends to stand the test of time. But there are a lot of things, there are a lot of caveats about that too. You know, does it degrade over time and so on. So you have to be careful with your interpretations. Yeah.
Looks like all your examples are fish that are something like 4 to 8 years old. Are any studies being done with much longer lived fish?
I wish I could get a lot of longer lived fish. I actually have one earstone from a fish from, it's another winter flounder from George's Bank. And I got it from the National Marine Fisheries Service. It was a 12-year-old fish. And it showed some really, really, really interesting things that all I can say is I've never seen anything like this. This fish, earstone does tell me the fish moved its habitat several times from sort of probably shallower, less saline habitat, nursery habitat, to deep water, cold water. But a lot of other things are in there too, all sorts of crazy rare earth elements. I'm still trying to sort it out. And then I've analyzed some old fish from Onondaga Lake. Onondaga Lake is the lake next to Syracuse. And that's, I mean, a whole lecture in itself about what's going on there. It used to hold the title of the country's most polluted lake. It's kind of cleaning up now. But I've seen very interesting patterns in other elements there. I have not looked at the manganese there, unfortunately, because we were still developing our analytical methods. So we didn't actually do manganese. We were looking at some other elements. I was actually trying to find mercury in the otoliths from Onondaga Lake fish because it's been heavily polluted with mercury from allied chemical and successors. So, but I didn't actually find mercury. I found selenium instead. So I had a whole nother project that's trying to figure out what that was about. Yeah.
You found a lot of gold in one of those. Does that mean that you could use that to find gold mining?
Oh, yeah, well, you wonder. I mean, I do find gold in marine otoliths, as it turns out. So this flounder, for example, from George's Bank had gold in it too, yeah. I'm not sure I'd go prospecting with it, but there are probably better ways to prospect. Yeah?
Is there a difference in the quantity of strontium that you're finding now in the air bones with the amount that you found in the Neolithic ones?
Yeah, it's a good question. And yes, I think actually thinking back to this work, we actually found, higher levels of strontium in relative to calcium in the Stone Age, these Neolithic ones. And I mean, on average, but they also always showed this sort of lower amount of strontium to calcium in the early years. And I think it's really sort of near, relatively near coastal nursery areas, and then moving out to deeper, colder water. But having said that, we do know from other studies, other geochemical and sort of piecing things together from paleontological studies, that the Baltic Sea was more saline at that time. It was. So it does seem to agree with the rest of our understanding. Yeah. It's pretty cool. You can see these things. Yeah.
Are there more questions for Dr. Lindberg? Well, let's thank her again.
Thank you.